Okay, so let's move with the presentation of Michele, non Riccardo. Michele Nodari, right?
No, Riccardo.
Sorry, Michele was the first one.
Yeah.
I expect a lot of curiosities from people because it's going to be a very interesting presentation. So he's Riccardo Nodari. He's finishing his second year of PhD at the University of Milan. And he's going to talk about paleomicrobiology and the study of ancient infectious diseases. So you have 15 minutes as well, and please, you can start with your presentation whenever you want.
Okay. Can you see the presentation?
Yes.
Okay, yes. Perfect. So, good afternoon, everyone. I'm Riccardo Nodari, a PhD student from the University of Milan. And today I'm going to talk about my PhD project, and in particular, I'm going to do a brief introduction into the field that I work on, which is not very well known, and it's the study of ancient infectious diseases. First, why we need to study ancient epidemics? And the answer to this is that studying ancient pathogens allow us to understand how they evolved, how they adapted to humans or to other hosts, how they spread geographically, or how their virulence change during their evolution. And all this information can be used to as a tool to work against current epidemics and also maybe prevent future ones. Until 30 years ago, the only way to study ancient epidemics was through an historical method, an historical approach, so studying text written by people that live during epidemic times. So this approach is very important, but have some major problems. First, if an epidemic was not recorded or all the information about it were lost, we cannot study it, of course. And other problems are the interpretation and the translation, and also the ability of the author of the time to properly describe what they see, the epidemics, the symptoms, and the effect on the people. The other approach to study ancient epidemics is paleomicrobiology, which is just a fancy word to say the study of microorganisms that are associated with ancient materials. So basically, what we do is to apply molecular biology, biochemistry, bioinformatics to research, to study molecules from ancient remains. These remains can be of many different types, and for humans, for example, we have bones, teeth, mummified tissues, but also paleofeces and others. And these, up to now, the paleomicrobiology is the only way to confirm a suspected diagnosis on ancient samples. But also in this case, we have some problems. The first one is the difficulty in finding suitable samples for the analysis. And even if we have these samples, well-preserved samples, all the molecules inside are going to be highly degraded and in low concentration. So the actual way to study ancient epidemics is to combine these two approaches. So the historical one to know where and what to search, and the paleomicrobiological one to give scientific proof of the presence of pathogens in the sample remains. I'm going to use plague as an example for the next slides because it's the focus of my project and also because it's the most studied ancient infectious disease. But most of the things that I'm going to show are also applicable to other infectious diseases. And so plague is an infectious disease that affects human and also other mammals. It is caused by a gram-negative bacterium called the Yersinia pestis, which was discovered in 1894 by Alexander Yersin. Here you can see him. Outside is a wonderful laboratory in Hong Kong, where he first detected Yersinia from a plague victim. And Yersinia pestis is responsible for 3 major pandemics in the last 2,000 years. The first one, the Justinian plague. The second one, which is the most known, that started with the Black Death in 1347 and devastated Europe for 400 years, killing half of the population of the continent. And also the third pandemic, which is still going on. In fact, the plague is not an eradicated disease, but there are not many, but some cases every year in states like United States, China, Mongolia, Russia, and also some major outbreaks. Here you can see schematic workflow for the analysis of ancient DNA. First, you need the samples, obviously, and you can have different samples, as I already said. In particular, I worked on teeth. These are some teeth from a suspected plague victim from an archaeological site in southern France. And why we use teeth is because inside them there is a soft tissue called dental pulp, which presents all the nerves and the blood vessels of the teeth. And after the death of the patient, of the individual, the teeth protect for thousands of years the dental pulp from outside contamination and environmental degradation. And since blood is circulating inside the dental pulp, we also have all the pathogens that circulate in the blood of the individual. To obtain the dental pulp, you need to cut the teeth. There are different ways to cut them, but one is to cut it longitudinally, so removing the dentin and the enamel from the middle of the teeth, and after that you have to open it and scratch the dental pulp, which is the brown reddish thing inside. And to scratch it, you can use a scalpel or an excavator, which is the tool that dentists use to remove tartar from the teeth. And here in the bottom, you can see the difference before and after extraction of the dental pulp. So when you collect the dental pulp, you can decide to extract the molecule of interest. And for example, for DNA, there are many commercially available kits that are used in forensic analysis or also modern DNA extraction. And basically the principle is the same. The only thing that it changes is the times that are usually longer, and also some steps must be repeated and other small things. When you know what you're searching for, historical information about your samples, you can decide to specifically search for a pathogen. For example, for Yersinia pestis, we search 2 genes, PLA and CAF1, which are specific for this pathogen. And after we detected the positive samples, we took these samples and we can do a genomic analysis, analyzing all the fragments of DNA that correspond to the organism of interest. And if you are lucky, you can also, and if you have enough material, you can also try to reconstruct a partial genome of the ancient organism of interest. For plague, we have now more than 80 genomes, and with these genomes, scientists were able to reconstruct a pretty detailed phylogenetic tree for Yersinia pestis with 2 major events. First, the differentiation, the speciation from Yersinia pseudotuberculosis, and then the acquisition of plasmids containing virulence genes that allow the Yersinia pestis to adapt to the flea vector. If you don't have historical information about the site or the individual, you can do, for example, a shotgun metagenomic to extract all the fragments inside and reconstruct the microbial community of your samples. And this technique is also used to reconstruct ancient microbiomes. In fact, we have multiple microbiomes from all kind of regions and time period. Also, we have a microbiome from the oral cavity of Neanderthal, dating back more than 40,000 years ago. And for the other approach, the historical one, I brought as an example a work that we did in collaboration with the University of Marseille. And this work, we adapted software normally used for in bioinformatics to analyze the gene expression, and we adapted it to analyze words inside ancient text. So we collected text written by people that assisted directly to two epidemics from the second pandemic, the Great Plague of Marseille and the Plague of Northern Italy of 1630, which is the famous plague epidemic that Alessandro Mazzoni made famous with I Promessi Sposi, and compare the words inside this text with the words inside control text, which are text written by people in the same time period, in the same location, but not talking about plague, but talking about other things like general medicine, agriculture, and sports, other things. The results of our analysis are actually here. So we have two word clouds for the words that are significative present in our plague-related text for French and Italy. And there are obviously the words associated with plague, like plague victims, quarantine, bubo, which is the most important symptom for bubonic plague, but also other interesting thing used for treatments or suspected sources of plague. Then we took these words of interest and we analyzed the connection with other words. So we took the words that are before and after these words of interest and generated a word network for the association between words. As an example, this is the complete network for the French dataset. Here are the network for the suspected sources of plague and other interesting words that express the effect of plague, like infected, contagious, mortality, and other. Here is the comparison between the two datasets for a specific word. So bubo, as I said, bubo is the typical symptom of bubonic plague, and in Italian is bubone. And you can see the words that are specifically associated with the Italian text, the French text, and in the middle, the words that are present in both datasets. And in this case, it's interesting to see that this symptom is located at the same part of the body, so the groin, the armpit, throat, in both epidemic.
Riccardo, you have 2 minutes?
Yeah, I'm almost finished. Other network for other words, infected or suspected sources of plague. And in conclusion, what we generated is a mostly unbiased method for the analysis of text, ancient text, that can be used in parallel with readings and interpretation of ancient text. And the specific for analysis on plague show that people of the time considered stuff like clothes, merchandise, and furniture as a potential sources of plague. And at the same time, the word rats is mentioned only 5 times total. Out of more than 1 million words, only 5 of these were the word rats, and never directly connected to the transmission of the infection of plague. And these results support the hypothesis that during the second plague pandemic, the major vector of Yersinia pestis was not rat flea, but human ectoparasites. I want to thank all the people that worked with me during the last 2 years, in particular my tutors, Professor Sara Epis and Professor Claudio Bandi, all my colleagues from the University of Milan, my colleagues from l'Ospedale Sacco, Francesco Comandatore, Professor Massimo Galli, and the Labanov group of Professor Cristina Cattaneo, and a particular thanks to Professor Didier Raoult and Professor Michel Drancourt and Remy Barbieri that hosted me the last year, basically all the last year in their laboratory and teach me most of the things that I know about this field. And thank you all for the attention.
Okay, thank you, Riccardo, for your nice presentation. I open the discussion if you have any questions. Alessandra has one question, I see. Please,
Hi, Riccardo.
Hi.
I promised I was going to ask you a question.
Yeah, I know.
I threw one away last week, but still I have another one. No, I wanted to ask you something because first of all, brilliant presentation. It was very interesting. Really, I think it's fascinating for all of us because we work on very different things. So I think it was very interesting. And then because you said you did an overview on the field and what can be done focusing on plug. I was wondering because you said your project is also on plug. I was just wondering what is that you do exactly? What's the aim of your project, or what do you work on the most?
Okay, so the project is actually multidisciplinary. As you saw, there is some historical analysis, like the one on the text from these epidemics, and the field work, which I don't go into the archaeological site, but I treat the samples when the anthropologist and the archaeologist gave them to me. So I don't go directly into the site, but we have a group of collaboration here in Marseille. I'm still in France, so the laboratory have some collaboration with many different archaeologists around France and also Europe. And also we have a collaboration with Professor Cristina Cattaneo and the Labanov group for the samples from Milan. And what I do is to search plague into samples from suspected plague victims.
Okay, and you said that you mostly use teeth in order to do that, because, and do you use any other samples, or you mostly use teeth because of all you said, because it's quite a complete sample and they keep DNA and pulp and blood?
Just for curiosity, I tried bones and compared with dental pulp, only for my curiosity, because it's known in the field that for infectious agents that circulate into the blood, the best sample that you can have is the dental pulp. So I tried also to scratch some bones, but the difference are evident and was only my curiosity.
Okay, so teeth were definitely better.
Yes, if you have teeth, you have to work on that because it's better. If you don't have them, you have to work on bones or what you have.
But even though you found a significant difference between teeth and bones, with the bones, did you manage to find the same results, even though maybe worse quality or stuff like that, or you couldn't get the result that you had with the teeth?
No, for only my test, so it was one time.
Okay,
I
just
find
okay.
no plague from the
Okay.
same individual that from the teeth I found marker of
Okay,
plague, but
okay,
the bones, no.
perfect. Thank you.
Welcome.
Okay, any other questions? No, I guess no. I wanted to ask the same question of Alessandra, so where did you get samples, but you already answered.
Sorry, too late.
It's okay, but it was a really nice presentation, so congratulations, Riccardo.
Thank you.